Hello, I'm Damla Senol Cali. Today, I will be presenting our work, GenASM, a low-power, memory-efficient, approximate string matching acceleration framework for genome sequence analysis. This work will appear in Micro 2020. Let's start with some background. The genetic material of an organism is called genome, and each genome consists of a DNA. And the information in DNA is encoded with a sequence of 4 different types of bases, A, C, G, and T. And genome sequencing is the process to determine the order of the DNA sequence in an organism's genome. And why do we care it? Because genome sequencing is pivotal in personalized medicine, outbreak tracing like COVID-19, evolution, and forensics. Because of the limitations of sequencing devices, whole genome of most organisms cannot be sequenced all at once, so first the genome is broken into smaller fragments. After each of these fragments is sequenced, we generate the small pieces of DNA sequences, which are called reads. Since we lose the location information of these reads with respect to the original genome sequence analysis requires first taking small DNA fragments from an organism and then reorganizing them into the entire genome. And success of all medical and genetic applications critically depends on the existence of computational techniques which will process and analyze the enormous amount of genomics data quickly and also accurately. However, effectively leveraging genome sequencing as a tool requires very high computational power, and also we need to process a large amount of data, so that's why we are bottlenecked by the current capabilities of computer systems. A common approach to perform genome sequencing is called read mapping, and read mapping is a method where we align reads against a reference genome in order to detect matches and variations. So we are identifying the original location using a reference genome. And this is one of the key components of genome sequence analysis. However, the bases in a read may not be identical to the bases in the reference genome at that original location due to 2 reasons. First, the sequencing technologies introduce errors into the read fragments during extraction. And second, the read may contain mutations or variations that are specific to the individual organism that the DNA was extracted from, which are not captured in the reference genome. So when we look at the left-hand side, we can see that we might observe 3 different types of errors. A base pair can be inserted, deleted, or substituted for another base pair. And in multiple steps of read mapping, we need to account for these errors. And due to their need to account for edits, multiple steps of read mapping are effectively a form of approximate string matching, or ASM, and can make use of existing ASM algorithms. Typically, ASM uses an expensive quadratic time dynamic programming algorithm, which accounts for over 70% of the execution time of read mapping and acts as the bottleneck for the entire genome analysis pipeline. So in this work, our goal is to accelerate ASM by designing a fast and flexible framework so that we can accelerate multiple steps of the pipeline. In order to replace the dynamic programming algorithm, in this work, we have focused on the Bitap algorithm because it performs ASM with fast and simple bitwise operations, which makes it highly amenable to acceleration. It is composed of 2 main steps. First, pre-processing. In this step, for each character in the alphabet, ACGT, we are generating 1 pattern bit mask. And these pattern bit masks indicate if character exists at each position of the pattern. And basically, we are translating the pattern into a bit vector based representation. And the second step, which is the main body of the algorithm, searching or edit distance calculation, here we compare all characters of the text with the pattern by using these pre-processed pattern bit masks along with a set of bit vectors that hold the status of the partial matches, and we only use bitwise operations in order to perform the calculation. I won't be able to go into the details of the algorithm due to the time limitation, so please refer to our paper for the details of the algorithm and its limitations. In this work, we propose GenASM, which is an ASM acceleration framework based on the Bitap algorithm. And in order to tackle the limitations of the algorithm, we optimize it, and also we design a new hardware. So we come up with a highly parallel Bitap algorithm, which has a very small memory footprint. And we also develop a novel bit factor-based algorithm to perform traceback, which is a critical step of read mapping, where we find the sequence of matches, substitutions, insertions, and deletions along with their positions, besides finding the mapping location with respect to the reference genome. And we design our accelerators as a processing in memory accelerators for both Bitap algorithm and the traceback algorithm. And we optimize GenASM for both short yet accurate and long but noisy reads. The reason is that different sequencing technologies produce different types of reads. So that's why we want to be comprehensive by supporting both of these scenarios. GenASM is composed of two main components, GenASM-DC and GenASM-TB. GenASM-DC generates bit factors and performs edit distance calculation. So DC comes from distance calculation. And GenASM-TB performs traceback, and TB comes from traceback, and assembles the optimal alignment. We also have DC SRAM for writing the intermediate data required for DC execution. And also we have a set of TB SRAMs for writing the bit factors generated during the DC execution, which will be later used in the TB execution. So our accelerators can be ported anywhere in the system, but in order to achieve high parallelism and low power consumption, we place our DC and TB accelerators along with the SRAMs in the logic layer of 3D stack memory. Our hardware is highly efficient in terms of both area and power, so we are within the budget limits of the logic layer. Let's look at the overall mechanism of GenASM. So first, read mapping starts, and the host CPU generates candidate locations in the reference genome for each read fragment. And for each candidate location, DC reads the corresponding reference genome region and the read from the memory and writes it into the DC SRAM. And after that, GenASM-DC starts searching for the pattern within the text region using the modified Bitap algorithm, and generates bit vectors and writes them into the TB SRAMs. And once DC completes its search, now GenASM will start reading the stored bit vectors from TB SRAMs, and it will generate the traceback output. Once TB generates the whole output for the full read, then it moves on to the next reference region and read pair. When we look at the hardware design of the DC component, we implement it as a linear cyclic systolic array in order to reduce the memory bandwidth and the memory footprint requirements. And processing core is the basic compute component, which is computing the bit vectors. And as you can see here, it has a very simple logic. And when we add the required logic, like flip-flops for data storage around the processing core, we come up with the processing elements. And multiple processing elements are concatenated to define the processing block. When we look at the TB hardware design, it also has a very simple logic, which is responsible for reading the bit vectors from one of the TB SRAMs based on the computed address, and then performing some simple, again, bitwise operations and some comparisons to find a traceback output for the current position. And by using these computations and comparisons, the third step is computing the TB SRAM address for the next position. And after it completes its whole execution, it writes the full traceback output to the main memory. We design our system around the 16 gigabytes of 3D stack HMC memory with 32 volts. And within each volt, we have a DC accelerator, TB accelerator, DC SRAM, and a set of TB SRAMs, so that we can obtain volt level parallelism by running each set of GenASM accelerators separately in each of these volts in parallel. In order to understand what are the use cases of GenASM, we need to look at the read mapping pipeline. So it can be seen as a 4-step process. First step, indexing. We index the reference genome, and this index contains all possible fixed length substrings as the keys and the exact match locations of these substrings in the reference genome as values. Second step, seeding, queries this index structure in order to determine the potential mapping locations of each read in the reference genome, and it generates the potential mapping locations. And in the third step, pre-alignment filtering examines the similarity between every read and its potential mapping segment in the reference genome. And the goal is to eliminate most of the dissimilar sequences to decrease the number of required alignments, which is the last step. And in the last step, for all the non-filtered candidate mapping locations, read alignment, or it's also called seed extension, step performs a dynamic programming-based alignment to determine the best location of the query read. And as part of this step, traceback operation between the reference segment and the query is also performed in order to find the optimal alignment. So except the seeding step, all of these three steps includes an ASM component. So that's why they are the natural use cases of GenASM. And read alignment, which is the fourth step, is one of the main use cases of GenASM. Pre-alignment filtering for short reads, which is the third step in order to filter out the dissimilar sequences, is also another use case. And on top of these, there is also a fundamental operation in genomics, which is finding the edit distance calculation between any two sequences in order to measure the similarity or distance between any two given sequences. So since GenASM-DC is inherently an edit distance calculation accelerator, this is our third use case. And in our paper, we also discuss other possible use cases of GenASM, including generic text search. Let's move on to evaluation. We model our design using a 3D stacked DRAM architecture, and we use two different data sets to show our applicability for both short and long read scenarios. And in this presentation, I will only show the details of our evaluation for the first use case, read alignment, and we compare GenASM against state-of-the-art software and hardware for this use case. When we look at our area and power numbers, when both accelerators operate at 1 gigahertz, and when we synthesize them using Synopsys Design Compiler, we observe that total power consumption for all volts is 3.24 watts for GenASM, and total area overhead is 10.69 millimeters square. For the first use case, when we look at the long read scenario, compared to the software baseline, one of the software baselines, Minimap2, we observe that GenASM achieves 116x speed up and 37x less power consumption. And compared to BWMM, which is another software baseline, GenASM achieves 648x speed up and 34x less power consumption. Compared to the state-of-the-art hardware for long read alignment, which is GACT of Darwin, GenASM achieves 3.8x better throughput while reducing the power consumption by 2.7x. And this trend is similar for short reads. Compared to the software baseline, GenASM provides significant speed up and reduction in power consumption. And compared to the state-of-the-art hardware for short read alignment, which is Scylla X of GenX, GenASM achieves 1.9x better throughput at a comparable area and power consumption. So please refer to our paper for the evaluation of other two use cases and also for more details. And to conclude, in this work, we focus on approximate string matching because it is one of the main bottlenecks of genome sequence analysis. And our goal is to provide an ASM acceleration framework in order to accelerate multiple steps of genome sequence analysis. And we propose the first work, which enhances and accelerates Bitap for ASM with genomic sequences. And we propose GenASM, which is a novel ASM acceleration framework where we co-design scalable and memory-efficient algorithms with low power and area-efficient hardware accelerators. And we have evaluated GenASM using 3 different use cases of ASM in genomics, and we show that GenASM is significantly more efficient for all the 3 use cases compared to the state-of-the-art software and hardware baselines. Thank you so much.